Welcome. Today we look at the metabolism of glucose. In the process of glycolysis. Glycolysis is simply a metabolic pathway that breaks down glucose, which is a 6 carbon molecule, into 2 molecules of pyruvate. Pyruvate is a 3 carbon molecule. So the 6 carbon molecule, which is glucose, is broken down into 2 molecules of pyruvate. And in the process, what is produced? Energy in form of what? ATP, and the molecules NADH, which are also used to produce ATP in the electron transport chain. That is glycolysis, and it occurs in the cytoplasm of what? Cells. It does not necessarily depend on oxygen for it to occur, but the amount of ATP that is produced is different in aerobic and anaerobic conditions. It is actually the first stage of what? Glucose metabolism, and provides energy when oxygen is absent or present. It also supplies intermediates for other pathways such as the fatty acid synthesis, amino acids, as well as nucleotides. That is glycolysis. Now let's look at the pathway. So the pathway is divided into 2 phases. There's what we call the energy investment phase and the payoff phase. In the energy investment phase, ATP is used up. ATP is used up. The payoff phase, ATP is regenerated. So first, the first reaction is that of glucose being converted to what? Glucose 6-phosphate. So you have glucose molecule, which is acted on by hexokinase or glucokinase, and this reaction requires ATP. Why? Because you need to phosphorylate the glucose molecule from glucose to glucose 6-phosphate. This is an irreversible reaction. So from glucose you generate glucose 6-phosphate. This reaction is catalyzed by the enzyme hexokinase or glucokinase, and this reaction uses what? ATP. So this is the first step in what? Glycolysis. In particular, the energy investment phase. As you can see, this is the first step that uses what? ATP. Then step 2 is just an isomerization in which glucose 6-phosphate is converted to fructose 6-phosphate. This reaction is catalyzed by what? Phosphoglucose isomerase, and as you can see, this is a reversible reaction. It's just a rearrangement of what? The carbon atoms of the molecule. Then fructose 6-phosphate acted upon by what? Phosphofructokinase 1. Phosphofructokinase 1. It also uses what? ATP to form fructose 1,6 bisphosphate. Fructose 1,6 bisphosphate. We've added another phosphate group to the fructose molecule. Actually, it should be noted that this is the rate limiting step. The phosphofructokinase is the regulatory enzyme, the most important regulatory enzyme. This reaction, the most important regulatory enzyme. This is another irreversible reaction. and it is actually the main regulatory step in the process of what? Glycolysis. Then fructose 1,6 bisphosphate is acted upon by the enzyme aldolase, which cleaves it into 3 carbon molecules of what? Glyceraldehyde 3 phosphate, glyceraldehyde 3 phosphate, as well as the dihydroxyacetone phosphate. Dihydroxyacetone phosphate. So these are the 2 compounds that are broken down. So at this point, from the formation of glucose, from the breakdown of glucose up to the formation of fructose 1,6 bisphosphate, this is what we call the energy investment phase, which leads to the breakdown of what? The 1 fructose 1,6 bisphosphate into 2 3-carbon compound molecules. The energy investment phase. As you can see here, ATP is used in the process at the first step and the third step. We're using ATP. Now, this dihydroxyacetone phosphate is then converted via triose phosphate isomerase to what? Glyceraldehyde 3 phosphate. This this guy here gets converted to what? Glyceraldehyde 3 phosphate for the process to actually continue. So as you can see, we're here. So from here, this is converted to glyceraldehyde 3 phosphate. Okay, maybe also let me also use this one. Yeah, dihydroxyacetone phosphate is converted to via the triose isomerase. Then you have now glyceraldehyde 3 phosphate, which requires the cofactor or the coenzyme NAD+ and converts it to NADH. So in the presence of what enzyme? Glyceraldehyde 3 phosphate dehydrogenase. This is reduced to what? 1,3 bisphosphor glycerate. This is converted to 1,3 bisphosphor glycerate, which is here. So glyceraldehyde 3 phosphate dehydrogenase converts the glyceraldehyde 3 phosphate into what? 1,3 bisphosphor glycerate. Then the 1,3 bisphosphor glycerate is converted to 3-phosphoglycerate by the enzyme phosphoglycerate kinase. Phosphoglycerate kinase. This reaction generates ATP. Yeah, as can be seen here. The conversion of 1,3 bisphosphoglycerate to what? 3-phosphoglycerate generates what? ATP, which is this reaction is catalyzed by the enzyme phosphoglycerate kinase. So it generates ATP. So it releases ADP to give off what? ATP. This step, the step or the process by which ATP is regenerated within the intermediates, and instead of using other sources, it is known as substrate level phosphorylation. The process by which ATP is regenerated just within the pathway itself is known as substrate level phosphorylation. Then from here, from 3-phosphoglycerate, it is again another somewhat rearrangement there, in which the phosphate group is shifted from position 3 to position 2, forming what? 2-phosphoglycerate. From 3-phosphoglycerate to 2-phosphoglycerate using the enzyme mutase. Using the enzyme mutase or phosphoglycerate or phosphoglyceromutase. So from 3-phosphoglycerate to 2-phosphoglycerate, we have what? Phosphoglycerate or phosphoglyceromutase. That is the enzyme there. Then there is what? Hydration that happens here. So with this hydration, water is added, and using the enzyme enolase, the 2-phosphoglycerate is converted to what? Phosphoenolpyruvate. Phosphoenolpyruvate. So water is added to the enolase enzyme, and which forms the phosphoenolpyruvate. The phosphoenolpyruvate is then converted to what? Pyruvate by the enzyme pyruvate kinase. In the process, there's a production of what? ATP. There's a production of ATP. So now remember that it is 2 ATP molecules that are what? Being produced. Because from this point, remember that there are 2 molecules of each of these. So because there are 2 molecules of each of these, it means even the ATPs that are being produced are 2, 2, 2, 2, 2. So at this point, ATP was produced. There were 2 ATP molecules that were produced. At this point, 2 molecules of ATP were produced. Again, this is another example of what we call substrate level phosphorylation. This is another example of what we call substrate level phosphorylation that happens here. So this is the 10 steps in the glycolytic pathway, divided into the energy investment phase up to this point, energy investment phase, and the energy payoff phase from this point all the way up to the formation of pyruvate. Now in the process, you notice that 2 ATP molecules were used up, then 4 of them were produced. 4 ATP molecules were done what? Produced. So the net means that if 2 are used up, 4 are produced, the net is what? 4 minus 2, which is what? 2. That is the amount of ATP that is produced. Of course, not forgetting that we also have what? 2 NADH molecules that can be also used by the electron transport chain to generate ATP. Yeah. Now, what are the fates of the pyruvate that is produced? So it depends on whether you have aerobic or anaerobic conditions. So in the presence of oxygen, pyruvate may be converted to acetyl-CoA via the PDH complex, and it will enter the citric acid cycle. In anaerobic conditions, if there's no oxygen, pyruvate may be converted to what? Lactate via the lactate dehydrogenase. Again, the fate of pyruvate depends on the ratio of, also apart from oxygen, it also depends on the ratio of NAD plus to NADH. This is what also determines on which path to take up the pyruvate. Yeah, so glucose, blah 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 blah. Then you have pyruvate. So pyruvate may be converted to acetyl-CoA and give off carbon dioxide in the process. Then also, like I mentioned, it depends on the ratio of NADH to NAD plus to NADH. So on one hand, it may be converted to acetyl-CoA, which may enter the tricarboxylic acid cycle. On the other hand, it may also be converted to oxaloacetate, which may also enter the tricarboxylic acid cycle. Then it may also be converted to ethanol and carbon dioxide, as well as lactate, especially in anaerobic conditions. Yeah, so that is those are the different fates of pyruvate, which depend on what? The presence or absence of oxygen, as well as the ratio of NAD plus to NADH. Now, what's the energy yield? So as earlier mentioned, you have in the investment phase, you have 2 ATP molecules that are used. Then in the payout phase, you have 4 ATPs that are produced. So the difference is what? The net, which is 2 ATPs that are produced. That is the energy yield. But again, remember that there are 2 molecules of what? 2 NADH molecules, and 2 and you know that these will give us approximately approximately 5 ATPs, because we know that 1 NADH is equals to what? 2.5 ATPs. 1 NADH gives us 2.5 ATPs. So it means if there are 2, then we get 5. So in total, we get about 7 ATPs. This is in aerobic conditions. In anaerobic conditions, it means that this NADH will not give us any ATPs because there's no oxygen to generate ATP via the electron transport chain. So what we may just have is what? The 2 ATP molecules that are coming from the glycolytic pathway. So we may just have 2 ATP molecules. So in aerobic conditions, you have more energy generated in form of ATP. In anaerobic conditions, there's less energy that is generated in form of what? ATP. Yeah, and this is just pointers to the regulatory steps. So as you can see there, the main and the final step, that is the conversion of phosphoenolpyruvate to pyruvate. Yeah, so the first step can be stimulated by what? The inorganic phosphate will stimulate the hexokinase enzyme. But at the same time, it can be inhibited by what? Glucose 6-phosphate. So meaning this one shows what? Product inhibition. So it is inhibited by the product it produces, glucose 6-phosphate. So if there's too much glucose 6-phosphate, it will inhibit this enzyme. But if there's too much inorganic phosphate, this enzyme will be activated to produce more glucose 6-phosphate. This can also go into the pentose phosphate pathway, another pathway that we'll look at on its own. Then you come now to the main regulatory step, which is the conversion of fructose 6-phosphate to fructose 1,6 bisphosphate, which is catalyzed by the enzyme phosphofructose kinase 1. What stimulates this enzyme, or what activates the phosphofructose kinase 1 is ADP and AMP. If there's an increase in ADP and AMP, this enzyme is stimulated. But what will inhibit it? What inhibits it is what? ATP and citrate. Too much citrate and too much ATP inhibits the enzyme phosphofructose kinase 1. That is this enzyme. The next regulatory step, the final step that converts phosphoenolpyruvate to pyruvate. This enzyme is inhibited by ATP. The pyruvate kinase 1 is inhibited by ATP, but stimulated by what? Fructose 1,6 bisphosphate stimulates this enzyme. Again, there are 3 regulatory steps, but the most important and the most important regulatory step, which is also considered to be the rate limiting step, is the conversion of what? Fructose 6-phosphate to fructose 1,6 bisphosphate, catalyzed by the enzyme phosphofructokinase 1, inhibited by ATP and citrate, but activated by what? ADP and AMP. They activate the enzyme. And that marks the end of this lesson. Thank you very much. I hope you enjoyed. Please don't forget to like, share, and subscribe. Cheers.